The next question. Autogamy can occur in a chesmogamous flower if. So what are chesmogamous flower? These are the flower which are like normal flower with exposed stamen and stigma. With exposed stamen and stigma. So Camelina, oxalis and viola plants produce two types of flowers, chesmogamous and clistogamous flower. So chesmogamous flowers are like normal flowers with exposed stamen and stigma, and in these flower chances of autogamy is less. Okay, so autogamy can occur in chesmogamous flower if pollen matures before the maturity of the ovule? No. Ovules mature before the maturity of the pollen? No. Both pollen and ovules mature simultaneously? So, both anthers and stigma are of equal length. All the three options are wrong. The right option is the third option, C. Both pollen and ovules should mature simultaneously. So these flowers have exposed stamens and stigma. There is more chance of cross-pollination rather autogamy. If in chesmogamous flower, the pollen release and receptivity of the stigma are synchronized, at that time there is chance of autogamy. So C is the option. Next question. Emasculated flowers have to be covered with a bag of suitable size, generally made up of. So during artificial hybridization, if a flower is a bisexual flower, we will remove the anthers or stamen carefully by using forceps, that is called emasculation. So emasculated flowers should be covered with a bag to avoid pollination from falling pollen grain. So with the bag suitable size, generally made up of butter paper. The option is butter paper, because polythene, nitrocellulose, these bags, they do not, rather than going for polythene and nitrocellulose, butter paper bags contains a minute openings and it is partially translucent in nature. So butter paper bags are covered. Next one. From the statements given below, choose the option that are true for a typical female gametophyte of a flowering plant. So they give the 4 statements. We have to choose the correct statement which is true for a typical female gametophyte of a flowering plant. The first option 1, statement 1, it is 8 nucleate and 7 celled at maturity. Yes. So the matured embryo sac is 7 celled and 8 nucleate structure, 8 nucleate and 7 celled. It is free nuclear during development. Yes, of course, yes. So single megaspore develops into embryo sac. This development is called monosporic. So during the embryo sac formation, which you have already understood, first free nuclear cell division leads to formation of 2 haploid nuclei. They move towards the opposite poles. Further 4 nuclei, 6 nuclei and 8 nuclei are formed. So these are all due to free nuclear division without immediate formation of cell wall. So it is free nuclear during development. That statement is also right. It is situated inside the integument but outside the nucellar. No. So where is the embryo sac present? Inside the nucellars, and this nucellars is covered by outer and inner integument. So this option is, this statement is not right. It's a wrong statement. It has egg apparatus situated at the chelazal end. No. Inside the embryo sac, the egg or female gamete is situated towards micropylar end, while antipodals are situated towards the chelazal end. So statement A and statement B are right one. Statement 3 and statement 4 are wrong one. So
The question is, from the statements given below, choose the option that are true for a typical female gametophyte of a flowering plant. So 1 and 4 are the right option. So option is 1 and 4 are the right statement, and the option is A. Next, match the following. They are given 2 columns. We should match them. So whenever there is a question, that is match the following type of question, always just draw a lines from column A to column B. Sporogenous tissue. Where do you find the sporogenous tissue? Inside the microsporangium. So before the anther is mature, the entire microsporangium is filled with homogeneous tissue called sporogenous tissue, and the cells of this sporogenous tissue later becomes pollen mother cell, PMC, and each of these pollen mother cell on meiotic cell division, they produce microspore tetrads. So sporogenous tissue is present inside the microsporangium. Nucellus. So nucellus is present inside the megasporangium or ovule. Male gametophyte. Reduced male gametophyte is pollen grain. It's not the gamete, it's a reduced male gametophyte. The spelling mistake is there. So consider as female gametophyte. So the female gametophyte is embryo sac. So A is 2, B 4, C 1 and D 3. So you check the option. Next question. Endosperm is not completely consumed by developing embryo in gram, bean, castor, pea. They are given. See, what is endosperm? Endosperm is a product of triple fusion. First, primary endosperm nucleus is formed. After double fertilization, triple fusion leads to formation of triploid primary endosperm nuclei. So I write it. So here this is the embryo sac. These are the synergids and egg together called egg apparatus. These are the 3 antipodal cells, and this is 2 polar nuclei present in the central cell. So one male gamete fuses with the egg to form diploid zygote, and the other male gamete fuses with the 2 polar nuclei to form triploid primary endosperm nuclei. So before the zygote develops into embryo, first a little endosperm is formed, and this endosperm is very much necessary for the nourishment of zygote, that is, which develops into embryo. So in some plants, even after the formation of the seed contain, they has retained the endosperm because the developing embryo has not consumed the entire endosperm. Such seeds in which still endosperm is found, that means the embryo has not consumed the entire endosperm, is called albuminous seeds or endospermous seeds. For endospermous seeds, the example is all monocotyledons. All monocotyledons, that is paddy seeds, wheat, maize, sorghum, jowar, etc. They are endospermous seed or albuminous seed because their seed as endosperm. But among monocots, orchid seeds are non-endospermous seeds. There's a one example among monocots which do not have endosperm, orchids. But in dicots, the developing embryo has consumed entire endosperm, so the seeds do not contain endosperm. Such seeds are called non-endospermous seed or non-albuminous seed. In all dicots, the seeds are non-endospermous or non-albuminous except castor. So castor is the plant
The botanical name is Resinus communis. And from this castor oil is extracted. So here what they have asked, endosperm is not completely consumed by developing an embryo in. It is consumed in bean, it is consumed in pea, it is consumed in gram. All are dicots. They are called non-endospermous nor non-albuminous seed. But among dicot, castor seeds do have endosperm. So it is endosperm. Endosperm is not completely consumed by the developing embryo in castor. So castor is the answer. Next one. Statement A. They have given two statements. Pollen grains of some plants remain viable for months. So in many plants, pollen grains have more viability time. They will be months together, especially Leguminaceae, Rosaceae members. Pollen grains in some plant remain viable for months. Yes, it's true. Double fertilization is absent where pollen grain shed in two cell stage. So whether the pollen grain get shed in two cell stage or three cell stage, double fertilization occurs. So let us go to the answers. There is options. Option A, statement B is correct and statement A is wrong. No. Both statement and A, B and D are no. Statement A is correct, statement B is wrong. No. Statement A is correct but B is wrong. Yes, this is the right option. C is the right option. Next question. Okay. Part of the endosperm remains liquid in coconut. Part of the endosperm that remains liquid in coconut is because. So I'll explain. So in coconut, immediately after double fertilization and triple fusion, the primary endosperm nuclei undergo repeated mitotic division, which is free nuclear without formation of cell wall, and this is called free nuclear endosperm. That is nothing but the coconut water what we see. It's called liquid endosperm or liquid syncytium. So after the free nuclear division leads to the formation of liquid endosperm, later cell wall deposition takes place. So this is what the carinal we see. The liquid is the product of free nuclear endosperm and this is the cellular endosperm. So the question is, part of the endosperm remains liquid in coconut because karyokinesis is not followed by cytokinesis. Option 2, karyokinesis is not followed by cytokinesis. Karyokinesis is followed by cytokinesis. Formation of liquid endosperm is not dependent upon karyokinesis and cytokinesis. None of the above. So you see the options. Karyokinesis is not followed by cytokinesis. So karyokinesis takes place and cytokinesis do not take place. This leads to the formation of liquid endosperm. So the option is the A. Next question. What would be the number of chromosomes in endosperm layer if megaspore cell contain 10 chromosomes? If megaspore mother cell, the point to be noted, what would be the number of chromosome in a endosperm layer if megaspore mother cell contain 10 chromosomes? They are given 4 options, 10, 20, 15 and 30. So these are very brain teasing questions. We have to work it out. See, inside the ovule there is a nucellus. A single cell present towards micropylar end of the nucellus develops into megaspore mother cell. which is diploid. It's a diploid cell. It's a part of nuclear cellus where one of the nuclear cell develops into megaspore, mother cell, which is a diploid cell. And this diploid megaspore mother cell undergo meiotic cell division. So it is referred as meiocyte. The cells which are capable of undergoing meiotic cell division are called meiocyte. This diploid megaspore mother cell undergo meiotic division, which leads to formation of 4 haploid megaspores. Right? Out of 4 haploid megaspores formed, 3 will get degenerated and only one will become functional megaspore. From this single functional megaspore cell, the female gametophyte or embryo sac is developed. So this is haploid functional megaspore cell. This haploid functional megaspore cell, what it will do? So it undergo free nuclear division to produce 2 haploid nucleus. Later, the free nuclear division leads to formation of 4 haploid nucleus. Further, it leads to formation of 8 haploid nucleus. So 4 are present towards one pole and 4 are present towards opposite pole. Later, this nucleus starts depositing the cell wall. So those 3 nucleus present towards the chelazal end, they becomes 3 haploid antipodal cells, and the 3 nucleus haploid nuclei formed from single functional haploid megaspore cell develops into 2 haploid synergids and 1 egg cell or female gamete together called egg apparatus. The 2 polar nuclei present at the haploid, they are also haploid polar nuclei present at the center. In the central cell, they are called polar nuclei or secondary nuclei. See here, all the 8 nucleus are haploid because they are derived from single functional haploid megaspore cell. So after fertilization, the egg N fuses with the haploid sperm to form diploid zygote. Likewise, 2 haploid polar nuclei fuses with the haploid male gamete to form triploid primary endosperm nuclei. So it is a triploid primary endosperm nuclei. Okay. Now let us come to a question again. What would be the number of chromosomes in endosperm layer if the megaspore mother cell contains 10 chromosomes? So if the megaspore mother cell contains 10 chromosomes, so out of 10 chromosomes, meiotic division leads to formation of 4 megaspore that containing 5 chromosomes. 3 megaspore degenerates and 1 become functional megaspore which is haploid containing 5 chromosomes. So on free nuclear division, all these 8 nucleus contain 5 chromosomes. Okay. So again, let us go to the question. What would be the chromosome number in the endosperm layer if the megaspore mother cell contains 10? So all the 8 nuclei are haploid. See here, an egg with 5 chromosomes, haploid egg with 5 chromosomes, fuses with a sperm belonging to same species, obviously the chromosome is 5. So to become diploid, that is 10 zygote. The zygote contains 10 chromosomes. In endosperm, 3 polar nuclei, 2 polar nuclei and 1 sperm fuses. Polar nuclei contain 5 chromosomes each, 2 polar nuclei, 10 chromosomes, and sperm 5 chromosomes. So the option is 50. So what would be the number of chromosomes in endosperm? Endosperm is a triploid. So layer, if the megaspore mother cell contains 10 chromosomes, so that is 50. So next question. Recognize the figure. The figure is embryo sac. So just carefully watch the figure. Let us recollect our knowledge. This is the embryo sac or female gametophyte. So embryo sac or female gametophyte. In this female gametophyte, this is the micropylar end. This is the chalazal end. So micropylar end consists of two haploid synergids and one egg cell or female gamete. Together they are called egg apparatus. Towards the chalazal end there are three cells which are called antipodals. Each cell is haploid because they are the product of meiotic cell division, and at the central large cell occupies two haploid nuclei which are called secondary nuclei or polar nuclei. So this is the structure of a mature embryo sac. Now let us go to the question. Recognize the figure given below and select the correct option. They have labeled A, B, C and D. So we have to mark the right one. So let us not see the options. Directly we can label them. A is nothing but antipodals. B is two polar nuclei. And C is the egg cell or female gamete. And D are the synergidae. So you check the option. So A is antipodals, antipodal. B is polar nuclei. Yes, right. And C is egg apparatus, egg, S, E, G, G. The spelling is mistake. And D is synergidae. So C is the option. Next question. During the formation of pollen grains, a microspore mother cell, that is also called pollen mother cell or microspore mother cell. So microspore mother cell undergo. So during the formation of pollen grain. So let us understand. So when we take a TS of anther, there are 4 microsporangium, 2 in each anther lobe, that is filled with a dense tissue called sporogenous tissue. As the anther mature, each of those sporogenous tissue develops into pollen mother cell, which is diploid. Pollen mother cell, and this pollen mother cell is also called as microspore mother cell. And this pollen mother cell undergo meiotic cell division. What is meiosis? A reductional cell division. 2n cell will produce on first meiotic cell division, and later on second meiotic cell division it produces 4 haploid. So 4 haploid, just a little up. So this diploid pollen mother cell on meiotic division produce microspore tetrads. So microspore tetrads, they are held 4 haploid cells each. They are called microspore tetrad. As they get dehydrated, they get separates into pollen grain. So the option is, during the formation of pollen grains, a microspore mother cell undergo 1 meiotic division, 1 mitotic division, 1 meiotic and 1 mitotic, 1 meiotic and 2 mitotic. No, the option is the first one. Only 1 meiotic cell division, which results in 4 haploid microspores. Next one. Select the correct and incorrect statements. Select the correct and incorrect statement. We should carefully read the statement. Tapetum nourishes developing pollen grain. Yes, of course, we have understood. Tapetum is the inner wall layer of the anther. So it's provide nourishment to the developing pollen grain. Helium represents the junction of the ovule and funicle. See, when we write the ovule structure, ovule covered by integuments, 1 or 2 layers. What we said, this is the body of the ovule. Body of the ovule is attached to the funicle at the region called ileum.
So at the region called ileum. So this is the ileum. So ileum represents the junction between the ovule. Junction between the ovule and funicle, that is called ileum. That's also right. So first two statements are the correct one. The third statement, let us check. In aquatic plants like water hyacinth and water lily, pollination is by water. No. So all aquatic plants do not show water pollination through water. So in water hyacinth and water lily, they have clearly mentioned they produce flower which emerges out of the water, and pollination takes place obviously by wind or by some insect. So here the pollination will not take, though they are aquatic plant, pollination will not take place through water. Okay. So the third statement is an incorrect statement. Fourth statement, primary endosperm nucleus is triploid. Yes, of course, primary endosperm nucleus is a triploid. It is a product of triple fusion. So let us check the options. 1, 2 correct. Third and fourth of statements are incorrect. 1, 2 and 3 third statement are correct. Again they will third incorrect. No. So first statement and fourth statement correct. 2 and 3 statement, second and third statement are incorrect. So let us check the option. The first and second statement and the third statement are correct. Here the option should be fourth statement. The fourth statement is incorrect. No, just I'll check. Sorry. So first statement, second statement, third statement are correct. Plus. Here they are not given the correct option. See, tapetum nourishes the developing pollen grains, right? Ileum represents the junction between ovule and funicle, right? In aquatic plants, water hyacinth and water lily, pollination is by water. Wrong. Primary endosperm nucleus is triploid. So check this option. Statement 1, statement 2, statement 1, statement 2 and statement 4, you make it 4, are correct. Third statement is incorrect. Next question. So one of the most resistant material, biological material. So the option is very, answer is easy. Lignin is a hard material present in sclerenchyma. Of course, we are studying in first year. Heavy cellulose. Along with cellulose, the cell wall of the plant cells are made up of heavy cellulose, lignocellulose, lignin plus cellulose, and sporopollen. There are 4 options. So, pollen grains consist of 2-layered wall, exine and intine. The outer wall is called exine, inner wall is called intine. And this exine is made up of a hard substance called sporopollenin, which is resistant to temperature, weak acids, weak alkalis, and even certain weak enzymes. So the option is sporopollenin. Third question. Fusion of one male gamete with egg and other of the same pollen tube with 2 polar nuclei. Fusion of one male gamete with egg and the other male gamete of the same pollen tube with 2 polar nuclei. So, when male gamete fuses with the female gamete called egg, it is called double fertilization. The second male gamete fuses with 2 haploid polar nuclei, that is called triple fusion. The answer is triple fusion. Next question. Endosperm of angiosperm is a product after fertilization of male gamete with endosperm is in angiosperm is a product after fertilization of the male gamete with secondary nuclei. Polar nuclei are also called as secondary nuclei. So central cell contains 2 haploid polar nuclei called secondary nuclei, with which one male gamete fuse to form a primary endosperm, which becomes endosperm. Next one.
Mark the following. So there is two columns, column 1 and 2. In column 1, A, B, C, D. And in column 2, 1, 2, 3, 4. Are the 4 mark the followings are there. So first one. Multicarpellary syncarpous pistule. Multicarpellary syncarpous, already we have understood papaver somniferum. Multicarpellary apocarpous pistule. This also rested Microspore champaka. One ovule in an ovary. One ovule in an ovary. So that's paddy. So these are wind pollinated plant. They generally contain single ovary in their ovule. Many ovules in one ovary. Orchids. So you check the options. So option they are not featured. So in the mark the following, just if you mark like this, that's enough. Then we can check the options. Multicarpellary, apocarpus, Michaelia. Okay, next one. Which of the following statements are true? They are given 4 statements. So they have asked the question, which of the following statements are true? The first statement, endothecium lies below the epidermis. Yes, of course. So the anther is made up of 4-layer wall. The outermost layer is the epidermis. Below the epidermis there is a layer called endothecium. Fusion of egg with male gamete is called apogamy. No, it's called syngamy. It's not apogamy. So first statement is true. Second statement is false. Synergids are applied. Obviously, yes, all the cells present inside the female gametophyte embryo sac are applied. Point at which funicle touches the ovule is micropyle. No, it's ileum. So, first statement and third statement are right. 1 and 3. So, 1 and 3. D is the option.